In this video, we will be using UCSF Chimera, a program for interactive visualization and analysis of molecular structures and related data. Specifically, we will be looking at our protein's three-dimensional structure in detail and using tools from Chimera, analyzing components of our protein. Then, we will look at deoxyhemoglobin S, a protein involved in sickle cell anemia, and mutating a residue using Chimera. First, ensure that you have installed UCSF Chimera. If you haven't, you can do so by visiting the link below. Next, go to uniprot.org. We will need to use our uniprot ID to grab the associated PDB ID. Do so by going to Retrieve ID Mapping. Type in your uniprot ID here. Then, going to Select Options, ensure that you're going from uniprot KB ACID to PDB, and hit Submit. Open Chimera. Under File, click on Fetch by ID. Ensure that PDB is selected, and input your ID. You should see the protein appear in the window, like so. Once loaded, there are different viewing controls. For example, under Actions, Atoms and Bonds, you can show or hide the atoms and bonds. Similarly, you can go to Actions, Ribbons to show and hide the ribbons, or Actions Surface to show or hide the surface representation, and Mesh to change the visualization from solid to mesh. You can also select which components will be represented. For example, to hide water, go to Select, Structure, and Solvent. And you'll see that you've now selected all the solvents in the protein structure. Next, go to Actions, Atoms and Bonds, and Hide to hide the selected atoms. We've now hidden all the waters. You can also change the color of the protein by going to Tools, Depiction, and Color Secondary Structures. In this case, we'll color the helices red and the strands purple. We can also set chains and residues to a rainbow color scheme by going to Tools, Depiction, Rainbow, and making sure Chain is selected. If we wanted to find all the hydrogen bonds, we could do so by going to Tools, Structure Analysis, and find hydrogen bonds. Ensure that Relax H-bond constraints is selected, and hit OK. To identify hydrogen bonds involving specific atoms, you can select the atoms involved by going to Select, Residue, and selecting one of the atoms. Then go to Tools, Structure Analysis, find hydrogen bonds, and select only find hydrogen bonds with at least one end selected. Once again, ensure that Relax H-bond constraints is checked. Hit OK. To identify residues within the vicinity of an atom, select Residue, and Ni. Then go to Select, Zone, and ensure that less than 5 angstroms from currently selected atoms is checked, and that Select all atoms and bonds of any residue is checked, and hit OK. We've now selected all the residues within 5 angstroms. To color them, go to Actions, Color, and select by element. If ever you wanted to manually measure the distance between two atoms, you can first select the residue, and then actions and focus to focus your view on that residue. Click on an atom by holding Control and left clicking it. Then pick a neighboring atom by holding Control and Shift and left clicking it. Go to Tools, Structure Analysis, and Distances. Then hit Create. Next, we will look at deoxyhemoglobin S and induce a residue mutation, changing a valine into a tryptophan. Start a new instance by going to File, Close Session. Fetch deoxyhemoglobin S PDB ID. The PDB ID for this is 2HBS. Select the residue under Favorites, Sequence, and select Chain H. This will show the entire Chain H amino acid sequence. You can mouse over letters to show residue numbers on the bottom. Click and drag the valine in the 6th position to highlight it in the visualization window. Now that we have it selected, you can select Action, Atoms and Bonds, Side Chain and Base, Show. Then, Actions, Focus to view the valine in the H chain. Then select Tools, Structure Editing, Rotamers, and change the rotamer type from valine to tryptophan. Hit OK. A new panel will appear showing all possible rotamer states of tryptophan. Each has a different probability associated with them. You can check for any clashes. Choose the rotamer where there appears to be a conflict. Hit OK. Select tryptophan by holding Control and clicking it. Then select atoms for clash detection by using the up and down arrow keys. Go to Tools, Surface Binding Analysis, Find Clashes and Contacts. Click on the Designate bar, which will show the number of atoms designated. Check All Other Atoms, and make sure that Select under Treatment of Clash and Contact Atoms is selected. Then hit Apply, but do not close the panel. Clashing atoms are highlighted in red, and a yellow pseudobond between them. To see how flexible that nearby threonine is in moving to accommodate the tryptophan, select both tryptophan and threonine 84. In my case, they are already selected. Go to Tools, Structure Editing, and Minimize Structure. Set the steepest descent steps to 15, conjugate gradient steps to 0, and the update interval to 3 so we can watch as it happens. Make sure that Fixed Atoms is unselected. Then go back to the Clashes window and uncheck the Select. And make sure to check After Relative Motions. Finally, go back to the Minimize Structure window and hit Minimize. Hit OK. Hit OK once again. And for the charges, hit OK once again. That concludes this video. You should now be familiar with navigating UniProt to access your protein's PDB ID and be familiar with Chimera's vast array of tools for sequence comparison and basic structure analysis.